Hi everybody, today we'll be focusing on the special senses, more specifically your sense of vision. So let's go ahead and talk about some of the main structures associated with your eye. This includes the eyebrows, eyelids, conjunctiva, lacrimal apparatus, external or extrinsic eye muscles that allows for the eye movement. Lacrimal apparatus is associated with the tear production and release of the tear from the eyes. So the proper word for eyelids is palpebrae, and there is a main muscle that allows the movement of the eyelid, referred to as the levator palpebrae superioris. We also have what we identify as conjunctiva, which is basically a translucent mucus lining that is present inside your eyelids, as well as covering the surface of the eye. If it's covering the inside of the eyelid, it's referred to as the palpebral conjunctiva, and if it's covering the surface of the eye, it's called the bulbar conjunctiva. And you can see it as this kind of a shiny layer that if you look at the surface of your eye, or if you look at underneath your eyelid, you can see that kind of glossy, shiny look to them, and that's basically that conjunctiva we're describing. If somebody has pink eye, that's an infection of the conjunctiva as a result of either a bacteria or viral infection. This is highly contagious, and people with pink eye should be avoided until the infection has been dealt with. Lacrimal apparatus is associated with your production of the tear. Lacrimal gland itself are located more laterally, present on both eyes, and they are connected to these ducts referred to as the excretory ducts that basically release the tear on the surface of the eye. Now, tears can move from the surface of the eye and reach the corner of the eye on an area referred to as the lacrimal puncta. If you guys recall from your skeletal system, there is a small bone on the side of the nasal cavity or nasal bone, referred to as the lacrimal bone, with a small hole in it called the lacrimal fossa or foramen. This tissue, lacrimal puncta, is sitting right on top of that. Lacrimal puncta allows the tears to empty into this duct system that we have called the canaliculi or lacrimal canaliculi, which empties to the lacrimal sac and then eventually going down the nasolacrimal duct and out through the nostrils. The structure of the apparatus or the lacrimal apparatus, the order of them or identification of the structure on the picture is something you guys should study for your exam. Next thing we have is the extrinsic eye muscles. As I mentioned, there are a total of 6 extrinsic eye muscles that basically give you the all the possible movements of the eye. And that includes moving the eye up or down toward the inner aspect or medially or laterally, or causing the rotation of the eye either through the movement more laterally or more medial movements. So let's kind of looking at some of the vocabularies we have. So when you think about the eye, if you're moving the eye laterally, it also can imply the movement is abduction as you're moving away from your midpoint. Versus when you move the eye medially, the proper word would be adduction. These words should sound familiar to you since we already discussed actions of the muscle in the muscular system during our lab. Superior rectus is, the other type of movements that your eye can make is elevation, basically moving up, versus depression, moving your eye down. Now you have a total of 6 muscles again. You have 4 of these muscles that are straight muscles, literally, and then if you think about them in the 90-degree angle from one another. You have the superior rectus, the medial rectus, the inferior rectus, and lateral rectus. And you have 2 on the side, one here and one here, that refer to as the superior oblique and inferior oblique. Now I'm going to demonstrate for you guys on a picture how these muscles actually cause the movement of the eye. So here is your eye, here's your pupil. Here is the first muscle. So if you kind of identifying the side, here is your medial side, here's the lateral, here's your superior, and here is your inferior. So the first muscle we have, which is located right here, is the superior rectus. So superior rectus is gonna move the eye up or elevate the eye and move the eye medially, which is adduction. So this is these arrows depicting your superior rectus movement. Medial rectus, as the name kind of implies, it moves your eye medially. So this is the easiest, one of the easier one to remember. So that's your medial rectus. The one that you have on the bottom, that's your inferior rectus. It moves your eye out, down and in also. So this is your inferior rectus. And again, it's moving your eye down and in, or it causes the depression of the eye, sorry, causes the depression and adduction of the eye. Lateral rectus on the side will be identified as moving your eye laterally. Again, simply looking at the name of the muscle can tell you the direction of the movement. Now, the other two muscles that we have are your superior oblique and inferior oblique. Superior oblique is gonna move your eye down and out, or depress your eyes and abduct your eye. So that's your superior oblique. And then inferior oblique is going to move your eye up and out. So that's your inferior oblique. So kind of think about the oblique, they're not straight, which means they're gonna do the exact opposite, I guess, if they're not straight. So inferior is gonna move the eye up as an elevate and move the eye out as an abduction. Versus superior oblique is gonna move the eye down or depress it and out or laterally, which is abduction. Now, there's two medical conditions associated with your extrinsic muscle. Diplopia or double vision is when the external muscles of the two eyes are not perfectly coordinated. To understand that when one eye moves laterally, the other one needs to move medially, right? Otherwise, the two eyes will be moving in opposite direction. When this happens, when somebody has double vision, because of the lack of coordination between the muscles, sometimes they end up with two separate images instead of one clear image, as the eyes are not moving in coordination. This could be a result of either a muscle of the extrinsic eye muscle being weakened. It might be a problem with the neurological component that controls these muscles, or it could be the paralysis of the muscle. Another problem with the extrinsic muscles of the eye is what we identify as a strabismus, which is also known as cross eye. This is a congenital disease or weakness of the eye muscles, where the one eye rotates either medially or laterally. In this scenario, the eye might take turn focusing on the objects, or only one eye is used to create an image. This can be problematic because brain starts to disregard the input from the deviant eye if there is a problem with that, which can eventually lead to a person identified as functionally blind. So there are, in the case of the cross eye, the person has to be treated, otherwise the issue would be basically for the rest of their lives. Now let's go ahead and take a look at the anatomy of the eye. So first thing I want to talk to you guys about in terms of anatomy of the eye is that the eye has three layers, and you can see them color-coded here. You have the outer layer of the eye, which is the thickest, and this is the fibrous tunic. And one word in terms of describing its function is protection. So kind of the goal is to make sure that the underlying tissues are protected. What is it made up of? It's made up the sclera, which is basically the white of your eye, and the cornea, which is the translucent bulging you see in front of the eye. Vascular tunic is kind of self-explanatory. It is going to contain a lot of blood vessels. It's providing nutrients for the eye, but it also contains structures like iris and ciliary body that controls the shape of your lens. And in this picture, even though it's not labeled, I'm gonna label this as your lens. And the third layer, which is depicted in yellow here, that's your sensory tunic, which is also known as retina. This is the one that's going to give you the ability to create basically vision. It is the layer that is the deepest layer and is highly protected. It does contain what we identify as pigmented layer and neural layer. Neural layer, to just kind of introduce the concept, it contains what we identify as photoreceptors that are basically responsible for creating your vision. So let's go ahead and get started with the three layers of the eye. The outermost layer is fibrous layer. We have the white of the eye as part of this layer, which is basically surrounding your eyeball. Or the outermost layer of the eyeball. This layer right here is your is your sclera. And interestingly, as the sclera comes in the front of the eye, it bulges out and becomes transparent because you basically want the light to be able to pass through it. It's still pretty thick. It still has an important role in terms of protection. However, cornea is very particular in two facts. First, it's identified as avascular. There is no blood vessels. So avascular implies no blood vessels. Why is this important? Because if you do have blood vessels on the surface of the cornea, basically your vision will be blurred. You won't be able to clear the perfect vision because you will continuously have something in front of the path of light. So where does your fibrous tunic receives its nutrients? It actually receives its nutrients from the fluid that flows in this space. This is a space called the anterior, and you'll see that in the future slide. It's called anterior segment, and it contains a fluid called the aqueous humor. And this fluid is basically where you get your nutrition. However, the oxygen that is required by the cornea actually directly comes from the air from your surrounding, which is, I think, is fantastic. Now, even though the eye does not contain, or your cornea does not contain any blood vessels, it does contain a significant number of nerves, a lot of these nerves being pain nerves, because again, your eye is extremely sensitive structure and you want to protect it to the maximum extent. The second layer, which you can see peeled off right here in that red color, that layer is your vascular tunic. Most of your vascular tunic is made up of the thick layer that basically lies right underneath the sclera of the eye, and that's referred to as the choroid region, or just simply choroid. Choroid is, again, highly vascularized. Lots of blood vessels is present here. This is basically supplying the nutrient and oxygen for majority of the structure inside the eye. A very important characteristic of the choroid. A lot of people assume that this layer is going to be reddish in color, even though if it's depicted as red in the picture, it's actually very dark in color because it contains a color pigment called melanin. Melanin comes from cells called melanocytes, so these are your cells. Melanin itself are proteins or color pigments that are basically give you your skin tone as well. So these dark pigments are present because you want to trap the extra light that comes into the into your eye, and preventing that light from scattering and affecting your image that will be created by your inner lining of the eye, which is your retina. Now, as you come in front of as you follow the choroid layer, so the red layer in this picture is your choroid layer right here, kind of peeled off, right? As you come here, you form this part of the structure right here called the ciliary body or ciliary muscle. So ciliary body or muscle consists of a smooth muscle, which makes sense again. It is something that's not under our control, but it is important in maintaining the shape of your lens and suspending the lens within the internal structure of the eye. Ciliary muscle is connected to these little strings that you see here, and these strings are basically what hold the eye or directly connect to the eye, and these strings are called suspensory ligaments. And again, they're holding the lens in position. So please understand as I go through this PowerPoint, if I contract or relax the ciliary body or muscle, I can pull the suspensory ligaments, and through that, I can change the shape of my lens. So imagine if you have a lens suspended in middle of a bunch of strings. If these strings are loosened, then there is no pressure on their lens. However, if the strings become, your suspensory ligaments become taut, then they're going to start to pull on either side of the lens, causing the lens to stretch, and that is extremely important for creation of the vision by focusing the light on the proper position. Here is again similar picture. This is an actual picture of the human eye dissected. So moving on, if you continue the eye, and I'm just going to go back to the original picture. If you continue the choroid layer and come in front of the eye, you find this structure right here, and this would be your lens. Sorry, this would be your iris. And right at the center of your iris, you do have a hole, and that hole is your pupil. So if you're looking at it at the frontal view, basically, again, this is your iris, and at the center of your iris will have a hole that is your pupil. Now, this should sound really familiar to you. We discussed it in terms of the sympathetic and parasympathetic nervous system in our previous lecture. Now, what are they exactly? Kind of reviewing the same information. Iris is identified as the color portion of the eye. It is made up of two types of muscle, the circular muscle and the radial muscle. So circular muscle is immediately surrounding the pupil, and the radial muscles are farther away or lateral to your circular muscles. So imagine this is your normal size of the pupil. Again, your radial and circular muscles are working together to form a specific opening or pupil size. Now, if you're dealing with a sympathetic stimulation, which is again would be your fight or flight response, the muscle that are involved is your radial muscles. Notice the arrows. Radial muscles are pulling, causing the pupil to dilate. So this is dilation of the pupil. Now, however, if you're dealing with a kind of a rest and digest, your parasympathetic nervous system, the main muscles that are engaged are the circular muscles. Notice again the arrows. These arrows are kind of showing the muscle contraction direction. So the pupil size becomes smaller, limiting the amount of light that comes through the eye. Basically, again, when you relax, you are limiting the passage of the light because your focus is not creating the best image. The third layer of the eye that we have is identified as sensory tunic or retina. And if I forgot, I don't remember if I mentioned that. I keep repeating the word tunic. Tunic means layer. So the tunic, the sensory tunic is made up of two layers. The outer pigment layer, and this layer is right next to your choroid, and the innermost lining, the deepest layer you have within your eye, that is identified as the inner neural layer. Now, what are the importance of these two layers? These two layers are important in terms of pigmented layer. They are very similar to choroid layer because they contain melanocytes, and their goal is to prevent light from being scattered. It is also a location for storage of vitamin A for your eye. The second layer that we have is the neural layer. Neural layer has three different types of neurons. First one, which is the most important one in terms of creation of your vision, are photoreceptors. Look at the number, guys. That blows my mind away every time I look at it. 250 million photoreceptors in your eyes. What is their goal? Create or ability to create vision, give you your ability to see. Now, photoreceptors are two types, cones and rods, and I'll talk about that in just a minute in more detail. You have bipolar cells, which are going to take the signal from the photoreceptors and move the signal alongside. Bipolar transmit their signal to the third category, which is the ganglion cells. Ganglion cells are facing the fluid that is inside of the car called the vitreous humor. And the axon of these neurons are merging together to form your optic nerve. So let me just show you the picture. So here is the three layers of the cell. And hopefully you guys can see that there's three different colors. You have kind of this purplish region, the reddish region, and let's say the greenish region. So the first layer you have, those are your photoreceptors. The one that is reddish in color, those are your second layer, which is your bipolar cells. And the third layer right here would be your ganglion cells. And this is where you will have your, vitreous, I can't spell, vitreous humor, where you have the fluid for the inner aspect of your eye, or what we identify as your posterior segment of the eye. So let's take a look at this again. If I'm looking at this layer right here, this as a whole, this picture as a whole would be depicting my neural layer. Neural layer is made up of what? Pigmented layer, and pigmented layer had a lot of what? Melanin, because it wants to prevent what? Light doesn't scatter, correct? Now, if I say what is on the layer that would be sitting right here, that would be your choroid layer, which is your second layer, right? Now, moving more internally into the eye, you have your photoreceptors, which pass the signal to the second layer, which is your bipolar cells. So if you want to label them on your picture, these are your bipolar. And then these cells right here in white are your ganglion cells. And notice how the axons of these ganglions are merging together. So these structures, when they all join each other, the axon of these cells becomes what we identify as your optic nerve, which basically gives you your image. Now, what I want to finish with is I want to finish by looking at the classification of your photoreceptors, which are your rods and cones. Rods and cones, what are the difference between them? So if you guys walk into a room that is relatively dark, you still can see where your objects are. You can see shades of gray, basically highlighting the location of the objects, but not necessarily any details. So the image that is created during such conditions are associated with your rods. Rods give you ability to see in dim light. Is there any detail? Is there any sharpness or vividness to the image created? The answer is no. Now, when you turn on the light as you walk into your house, everything becomes clear, right? You get a vivid image, you have beautiful, sharp images with a lot of colors. Your ability to create that is associated with the second category of photoreceptors known as cones. Cones are important because they give you the ability, again, to see colors and detailed quality images in the presence of light. There are three different types of cones, blue, red, and green, with basically sensitivity to blue color and red color and green color. The other variation in terms of color and shades that we see is by activating different numbers of these three different type of cones that basically, again, give us the different shapes and colors that we will see on a daily basis. Now, again, kind of going over this, same information, rods are vision in a dim light or peripheral vision. They are identified as more sensitive, and this is the one that my students kind of trip over sometimes. So rods are in a dark, so how are they sensitive? Think about this. When you are in a dim environment, do you have a lot of light present? The answer is no. But those photoreceptors only use that very little light to get activated and create an image, which means very little light means those cells are extremely sensitive to light because you only need very little light to activate them. On the other side, cones are identified as what? As least sensitive to light. So these are the ones that require a significant amount of light for them to go and to become activated and create your image. Again, same concept, just in terms of pictures, low acuity versus high acuity for your cones versus your rods. If I ask you which one is more numerous, your rods are definitely more numerous. There's about 20 rods for every cone. And your rods are more in the peripheral aspect of your vision versus your retina, where you're focusing on the image right in front of you, that will contain more cones. Now, photoreceptors, the location where all of your image is actually going to focus is an area called fovea centralis. It contains all cones with no rods, and it's going to give you your best visual acuity. So when you do eye movement, it allows for you to focus on objects at the fovea centralis, again, give you the best image that you can create for your surrounding. Couple last things about this. We already talked about bipolar and ganglion cells. It just highlights the same information. Photoreceptors are the one that creates the original signal, and that signal gets passed on to the bipolar, and bipolar pass that signal to your ganglion cells. Axons of these ganglion cells are all merged together and come out of the back of the eye as the optic nerve. And hopefully you remember the optic nerve eventually move its signal all the way to the occipital lobe of your cerebrum, allowing you to create your sense of vision. Now, we also have something called the optic disc, not to be mistaken with the optic nerve. Optic disc is the site where the optic nerve leaves the cell, it leaves the eye. So right here is where your optic nerve is leaving, therefore making this your optic disc. Because this is where all the axons are leaving your eye, then there is really no room for any photoreceptors. And because there is no photoreceptors, it means you cannot create an image or focus light on that spot. Therefore, your identifies as your blind spot. Here's a picture kind of highlighting your optic disc, where the optic nerve exits. And this is another area you can see is a darker color called maculatua, which is basically where you will have the highest concentration of your cones, giving you your visual acuity, better known as your fovea centralis. We have a medical condition called retinal detachment. This is where your pigmented layer, which is the second layer of the neural layer, gets separated. Sorry, the second layer of the sensory tunic get separated from the neural layer, which is where your photoreceptors are. This detachment basically allows the vitreous humor fluid to flow between these two layers and allow the separation of these two layers. And when these layers separate from each other, basically, you lose the ability to provide nourishment for your photoreceptors. No nourishment, your cells start to die off, and as a result, that can cause to permanent blindness. Who is more susceptible to developing retinal detachment is when people who do bungee jumping or have been exposed to severe trauma, where you are moving and all of a sudden you stop, and that pressure can literally separate these two layers from each other. Okay, I'm going to stop here and then we'll continue this PowerPoint and finish up the phototransduction in our part 2 of sensory lecture.